Hello students, in today's class we are going to learn about next generation sequencing reaction. The principle behind next generation sequencing is similar to that of the Sanger sequencing, which relies on capillary electrophoresis. The genomic DNA is first fragmented and the bases in each fragment are identified by the emitted signals when the fragments are ligated against a template strand. The next generation sequencing method uses array-based sequencing, which combines a technique developed by Sanger sequencing in order to process millions of reactions in parallel, resulting in a very high speed and throughput sequencing at a very reduced cost. There are basically three steps to next generation sequencing: library preparation, amplification, and the actual sequencing step. So libraries are created using random fragments of DNA followed by ligation with custom linkers. Amplification step, the library is amplified using clonal amplification method and PCR. The third step, that is sequencing, for which we make use of one of the several different approaches. Coming to the first step, that is library preparation. So first the DNA is fragmented either enzymatically or sonication method to create smaller fragments of DNA. Then adapter molecules are ligated to these fragments with the help of enzyme DNA ligase, and the adapters basically enable the sequence to become bound to the complementary sequences known as primers, which are attached onto the surface of a chip that is used for sequencing. Use of adapters could lead to potential problems of base pairing between the molecules and therefore dimer formation. And to prevent this, the adapter molecules are chemically modified so as to remove the 5' phosphate group, and this leads to generation of hydroxyl group at both ends, and hence it will allow ligation only at one end to the insert molecule. By removing the phosphate from one end of the adapter and therefore by creating 5' hydroxyl end, the DNA ligase is able to form a bridge between the two terminals. So here is a diagrammatic representation of the process. So first you need to have your genomic DNA, which is fragmented by different methods. So you are going to have smaller fragments of double-stranded DNA. The next step is attachment of adapter molecule. The adapter molecule is dephosphorylated at one end so that you're going to have hydroxyl groups present at one end. The other end contains hydroxyl and phosphate groups both. And then you mix your insert molecules with the adapter molecule in presence of enzyme DNA ligase. And these adapters are going to get ligated at both ends of your insert molecules. Now this is going to result in formation of a library. So library here is this collection of these different fragments, and to each end of the fragment you have attachment of the adapter molecules. Here the two strands of the adapter molecules are represented in different colors. So these two strands are actually complementary to each other. So you can see them attached at either ends of the DNA fragment. Now, in order for the sequencing reaction to be successful, the library fragments need to be specially clustered in the PCR colonies, as they are conventionally known, which consist of many copies of a particular library fragment. Since these colonies are attached in a planar fashion, the feature of an array can be manipulated enzymatically in parallel. This method of library construction is much faster than the conventional methods, which are labor-intensive methods, in which we clone the fragments into the vector and transform the bacterial cells, and we pick up individual colonies to do the screening, as well as then use it for amplification and Sanger sequencing method. This is very time consuming method, and it is not possible to do whole genome sequencing by this kind of approach. So the second step is amplification. So library amplification is required so that the received signal from the sequencer is strong enough. So initially what we have is like one fragment of DNA to the either end of which the adapter molecules are attached. So we need to convert this one fragment of DNA into multiple fragments, which is basically known as a DNA cluster. So each DNA cluster is formed from each DNA fragment. So this is required so that we can have a strong signal during the sequencing reaction. So different types of two types of methods you can say are commonly used for the clonal propagation. One is known as an immersion PCR method. In immersion PCR method, we make use of immersion oil, beads, and all the components that are required for the PCR and the library DNA molecules. So they are all mixed and they are aliquoted into different wells. These wells are known as micro wells. Here we need automation for the process. Now, what will happen is we must have in a particular well, we must have a single bead and a single DNA fragment for the success of the method. Now we'll add all the components that are required for multiplication, and what will happen is that these DNA fragments are going to attach onto the surface of the primers that are present on the surface of the bead. So on the surface of the beads, there are actually two types of primers, as we had seen the two ends of DNA. So both the ends of the DNA are able to bind to the two different types of primers that are attached on the surface of the bead. Although in the diagram we can see only one color, but actually you have two different types of primers that are present. So these DNA fragments will attach onto the primers, and then the extension reaction will take place, which will actually result in multiplication of the fragments. At the end of the process, you are going to have one fragment converted into many copies of the fragments attached to the surface of the bead. So these points are written in the slide for your reference. Also, the second slide for your reference on clonal multiplication by emulsion PCR. So this method overall requires about 30 to 60 cycles, leading to generation of clusters of DNA. The other method, uh, this is again another slide for your reference. And next we'll talk about the other method that is known as bridge PCR. Okay, so in bridge PCR, instead of having the bead-like structures, what we have is basically a chip, and the chip is divided into smaller portions which are known as cells. Okay, so what we do is also on the surface of the cells present on the chip are present these primers. So as you can see, there are two types of primers. The one in red, that is complementary to the blue portion of the adapters, and the one in blue that is complementary to the red portion of the adapters. So here what we do is we first need to have the chip on which the primers are basically immobilized, and then we add DNA on the surface of the chip. So the dilution of the DNA should be appropriate so that each DNA fragment is going to attach to different cells on the surface of the chip. So after that, since these are single stranded fragments, these are going to fold upon itself and bind to the complementary primers by the complementary base pairing at the other end. And after that, we add all the reagents that are required for PCR, and this is going to convert the single stranded DNA into double strand. And after that, we do a denaturation step. So the two strands are going to get separated. Again, it will bend down and form a base pairing with the complementary primers that are attached. And in this fashion, the single DNA fragment originally that is representing one fragment of the library is going to multiply to form clusters of identical strands, which is known as clonal propagation or PCR amplification on the surface of the chip that contains several cells. Attached to each cell is present one DNA fragment that gets amplified to form a cluster of that DNA fragment. So the steps are mentioned here for your reference in the first and the second slide about bridge PCR. Now coming to the third step, that is sequencing. Okay, so sequencing, several competing methods of next generation sequencing have been developed by different companies, and here we are going to discuss about two types of methods. One is the 454 pyro sequencing, and the other is reversible terminator sequencing that makes use of two types of terminators, 3' O block reversible terminators and 3' unblocked reversible terminators. So coming to the pyro sequencing. So pyro sequencing is based on sequencing by synthesis principle where a complementary strand is synthesized in presence of polymerase enzyme. Now, in contrast to using dideoxynucleotides to terminate the chain amplification, pyrosequencing reaction basically detects the release of pyrophosphate molecule when the nucleotide is added to a DNA chain during extension. So here we have a diagrammatic representation of the process. This is the template DNA. We add the single primer for extension reaction. Only one primer is added. And we add one of the four DNTPs at a time. So we add the four DNTPs in a sequential manner to flow over the surface of the colonies. Okay. Now, when the particular base is incorporated, that will result in release of a pyrophosphate molecule. This pyrophosphate molecule is converted into ATP in presence of ATP sulfurylase and adenosine. And this ATP molecule is used up by enzyme luciferase that will convert the substrate luciferin into oxyluciferin, and along with that you will have emission of light. The same steps are explained here. Also, the relative intensity of the light is proportional to the amount of the base added. So suppose if you have two adjacent same types of nucleotides present on the template, then in response the complementary two bases will be added. Okay, so this is going to release twice the amount of pyrophosphate, and hence eventually twice the amount of light intensity will be released, and hence twice the amount of signal is detected by the machine. So these are some points for your reference related to pyrosequencing. Now coming to the next method that makes use of reversible terminator sequencing. So reversible terminator sequencing method differs from traditional Sanger method in that instead of terminating the primer extension irreversibly by making use of dideoxynucleotide, we make use of a modified nucleotide, and these cause reversible termination reaction. So here is a diagrammatic representation of the process. So the mechanism uses sequencing by synthesis approach, elongating the primer in a stepwise manner. So firstly, the template needs to be immobilized or fixed onto a solid support, which is already done in the previous step. And then the support is we add primer and everything that is required for PCR, and we add all the four bases which have four different fluorophore groups attached to the base, and in addition they show or they have addition of a azidomethyl group at the 3' hydroxyl end. Now in each cell, depending on the template, the correct piece will be added, and it will give a different type of a signal. Then you do a washing step to remove whatever excess nucleotides are present, and after that Once the correct base has been annealed to the target, we do a treatment with a chemical which is known as TCEP, which stands for tris 2 carboxylethyl phosphine. And this basically removes the fluorescent branch as well as it removes the 3' O-linked azidomethyl group. And once the azidomethyl group is being removed, that regenerates the 3' hydroxyl group. And now after that, you can add another round of nucleotides is added. And again, after polymerization, you will have addition of the nucleotide, and then again the excess nucleotides are washed out, and the signal is recorded. Again, the process is repeated. So here, every time you have to wash off the excess bases, and enzyme also needs to be added every time. Then coming to the 3' unblocked reversible terminators. So these are different from the 3' blocked reversible terminators, in which in the previous case the 3' hydroxyl group was blocked by a chemical, and here it is not blocked. So the reversible terminator group of the 3' unblocked reversible terminator is linked to both the base and the fluorescent group, which now acts as a part of the termination group as well as a reporter. This method differs from the 3' O block reversible terminators in three ways. Firstly, the 3' position is not blocked, and the fluorophore for all the four bases is the same type of a fluorophore. And here, each modified base is flowed in a sequential fashion rather than at the same time. So this is about next generation sequencing. Kindly like, share, and subscribe if you like the contents. Thank you.